Hello everyone, welcome back to our Plantae. In today's lecture, we'll learn all about the life cycle of angiosperms in a simple and easy to understand way. So, let's begin the life cycle of angiosperms. Life cycle of an angiosperm comprises of two generations, the haplophytic and the transmorphic. These two generations alternate with each other in regular sequence. This phenomenon is known to follow alternation of generations. The sporophytic plant, that is usually differentiated into roots, stem, leaves and flowers, is the sporophyte. It is the dominant phase of the life cycle. It consists of diploid cells, 2N. It is the site of reduction division. It produces haploid microspores and megaspores. The haploid phase, gametophytic phase, is represented by two structures, the male gametophyte and the female gametophyte. The male gametophyte is represented by pollen grains and the pollen tube with two sperm nuclei and a tube nucleus. The female gametophyte is generally represented by 8-nucleate embryo sac. Both gametophytes are highly reduced and completely dependent on the sporophyte. Diagrammatic life cycle of an angiosperm plant. Do you need help understanding any part of the life cycle of angiosperms, or want me to explain something specific from this chapter? During the course of evolution, there has been gradual reduction and loss of independence in gametophytic generation, whereas sporophytic generation has registered gradual increase in size to attain even tree-like proportions in angiosperms. The oospore, thick-walled zygote, produces embryo by segmentation. The ovule produces seed and the ovary the fruit. The seed and fruit protect the embryo and also store food for it. The seed germinates into a seedling and then slowly develops into a mature plant. Development of flower. The flower is a modified shoot and appears as a small bud first and develops much like a foliage bud. However, it differs from foliage bud in that there is no elongation of the internodes. Therefore, the floral leaves are grouped together and not distributed at intervals as leaves on stem branches. The primordium of flower appears as a small axis of meristematic cells, which enlarges into a rounded structure, the receptacle, and a narrow stalk, which becomes pedicel later on. The floral parts arise at the tip of the receptacle as rounded protuberances of meristematic tissue. At 4 points, the circles of 4 sepals appear first on the receptacle, shepherd's purse. These are followed by primordia of stamens. The pistil arises as a continuous wall about the apex of the floral axis. The formation of pistil primordium stops growth in length of the floral axis. The petals appear in the last. The development sequence of different parts of the flower varies in different species. For example, in buttercup, the different floral whorls appear spirally on an elongated receptacle, acropetally. The sepals coming first, next the petals, then the stamens, and finally the carpels. In members of family Compositae, the sepals appear last, remain vestigial, and later on transformed into pappus. In Umbelliferae, the sequence is stamens, petals. Sepals and pistil. Different stages showing development of flower. Microsporophyll or stamen. Each stamen consists of 3 parts, i.e. filament, anther, and connective. The filament is the stalk of the stamen, and at its tip 2 anthers connected with a sort of midrib called connective are present. Each lobe of the anther contains 2 microsporangia or pollen sacs. Thus, there are 4 chambers in each anther. Each pollen sac contains mass of cells in pollen grains or microspores, A cross-section of young anther shows that each anther lobe contains 2 pollen sacs. Number may vary from 1 to many. As the anther matures, the tissue between each pair of pollen sacs and the 2 pollen sacs disintegrate, and 2 large cavities are produced instead of 4 in each anther. Within each microsporangium, a number of microspore mother cells are produced, which is surrounded by a layer of nutritive cells, the tapetum. Outside the tapetum, many layered wall of the sporangium is present. The outermost layer, beneath the epidermis, shows characteristic band-like fibrous thickenings. This layer is endothecium, which helps in dehiscence of anther, opposite the partition between each pair of pollen sacs. The cells of the endothecium are thin-walled and form the stomium, which marks opening of ripe anther. Each microspore mother cell undergoes meiosis to produce 4 microspores. Development of microsporophyll. Each microsporophyll, stamen, arises as a small papillate outgrowth of meristematic tissue from the growing tip of the floral primordium. It grows actively and is differentiated into an apical broader portion, the anther, and a lower slender part, the filament. The transverse section of a very young anther shows that it consists of a mass of meristematic cells surrounded by an epidermal layer. Development of microsporangium. In each lobe, a group of hypodermal cells becomes differentiated from the neighboring cells by enlarging in size, having dense cytoplasm and prominent nuclei. These are the sporangial initials, which generally consist of a 2 to 3 cells wide plate, running along the entire length of the lobe. These cells divide periclinally, forming a primary parietal layer and a primary sporogenous layer. The cells of the primary parietal layer lie beneath the epidermis, and by periclinal and anticlinal divisions give rise to 3 to 5 concentric layers of cells. If you found this video helpful, please give it a thumbs up, share it with your friends, and don't forget to subscribe for more easy biology lectures. Also add 3 questions at the end of lecture. Thanks for watching, and see you in the next class.